so let's now proceed to bacterial contamination in meat. Again, this is just a review of some things we discussed in the past and maybe your general or food microbiology, no? Excuse me. Recall that muscle meat is generally sterile, but again, microorganisms are found in the skin, hooves, and innards, specifically the intestines of the animals. And these things could, during slaughter and post-slaughter process, leach into the other sterile parts of the meat. So generally, a good microbiological standard for aerobic plate count or total plate count is less than 10 to the 4 for fresh meat. And for Enterobacteriaceae, that's less than 100 or per square centimeter, no? So surface swabbing ito, no? Okay, and then of course, between 4 and 5 log CFU yung critical, and then not acceptable for greater than 5 for APC, and for Enterobacteriaceae, that's between 100 to 1000 critical, and greater than 1000 yung not acceptable. So alam naman natin, again, recall na the APC is an indicator of general hygiene. It doesn't necessarily mean na may pathogens doon. But of course, it can also tell you kasi above certain APC, like ba nakita natin mga 10 to the 6, 10 to the 8, pwede ka na magkaroon ng mga quality indicators like yung sliminess, kakaibang amoy, etc. And Enterobacteriaceae, of course, we know, are indicative of fecal contamination in some cases. Okay? That temperature is a great factor in growth of microorganisms storage. So in this case, as you can see here, there are two major factors determining the shelf life of raw meat in storage. It's the initial microbial load and the storage temperature. Take note na ito yung critical region na nakita natin sa previous slide, no? Yung 10 raised to 4 and 10 raised to 5. Definitely, if you have a higher microbial load to start with, then i-ikli yung shelf life niya by a few days. And as you can see here, typical refrigerator sa Pilipinas, although dapat nasa 0 to 7 degrees yan, normal po yung ref na umaabot ng 10 degrees, lalo na pag summer, no? Lalo na yung mga ref na wala namang temperature indicator. Okay? So as you can see, talagang napaka-ikli ng shelf life kapag nag-refrigerate ka ng meat. Let's say yung pinapag-defrost mo siya at refrigeration temperatures na either around 5 to 10 degrees. So definitely that already shortens the shelf life of your raw meat. You need to move as fast as possible. And again, if nakabili ka ng karne halimbawa sa wet market, at to start with, questionable na yung microbial load niya, unless you put it in a freezer, you really need to cook that right away, no? Right, so if we remember correctly, yung pagkain natin, merong dalawang paraan para usually siya ay either maging unsafe kainin or hindi na kaaya-ayang kainin. The first is through spoilage microorganisms. So, meat spoilage typically involves yung spoilage microorganisms natin, usually mga aerobic plate count results yung nag-determine nito. And may kaakibat sila usually na mga sensory changes in meat, no? So we have putrefaction, ayan, tama siya, magbabago yung amoy niya, and that's caused by a breakdown of protein caused by various microorganisms. There's also slime formation na paghawak mo parang yak na feeling slimy. Okay? That's also related to an increase in spoilage microorganisms. May include a change in color and density caused by breakdown of fats, with a few microorganisms that can break down fats. Next is, of course, when we have pathogenic microorganisms, if you recall from your general or food microbiology, when we have pathogens and they are allowed to grow, they can they not necessarily change yung sensory properties ng meat, but they now pose danger once ma-ingest natin yung food through the two basic pathways of infection and intoxication. Again, as you recall, when we say infection, yung pagkain ay contaminated ng microorganisms and doon sa loob ng katawan mo after mong kainin, doon magpo-produce ng toxic substances yung microorganisms. So to prevent infections, of course, you have to kill the target microorganisms through usually heat treatment or another form of antimicrobial, no? Intoxication, on the other hand, involves the pathogens growing enough in meat so that before mo pa siyang i-ingest, nakaproduce na sila ng toxins. Ang nakakatakot dito, once the microorganisms have produced toxins while inside the food, even if you kill the microorganisms, the toxins will remain. And therefore, usually kapag may toxin na dyan sa pagkain mo, kahit ano pang thermal treatment mo dyan, hindi niya kayang tanggalin yon to a safe level, no? Kasi usually kapag mga toxins, hindi naman yan namamatay sa boiling or yung chemical structure nila hindi na disintegrate at lower temperatures. No? So, yun yung nakakatakot sa infection. And again, when you have pathogens or food poisoning, this does not necessarily mean na amoy panis, lasang panis yung food. They could look or smell, taste perfectly fine. But well, yeah, you get the point. It could kill you without knowing it, the worst case scenario. Okay, so looking at the microorganisms responsible for most of these college mediators, you can refer to this handy table. Of course, this is not the end all and be all. Pero generally, ito yung mga involved. No? So, putrefaction, which is involved with protein breakdown, commonly attributed to Pseudomonas and other cold room flora that grow in lower temperatures, as well as those from the Proteus and Clostridium genus. For souring, of course, obviously yan yung mga nagpo-produce ng acids like the Lactobacillus enterococcus and Pediococcus, collectively referred here on forward as the lactic acid bacteria. So, hindi lang lactobacillus, yung lactic acid bacteria natin. For fermentation, then obviously there are yeast, Enterobacteriaceae, and certain LABs, which of course obviously cause fermentation to produce acid. Meanwhile, of course, the yeast, alam naman natin, they ferment to produce other things, carbon dioxide. And of course, in certain cases, we also have fermentation, but not so much in meat. For turbidity, again, Enterobacteriaceae and LABs, quickly discoloration is also caused by LABs. On the other hand, for slime formation, pretty much halos silang lahat, plus C-streptococcus. And again, there's a distinction on whether it's primarily happening under slime formation under the presence of much oxygen or kung vacuum packed, or if it's in fermented meats, no? Kasi depende nga kung ano yung condition ng oxygen and pH ng meat mo, kung ano yung mag-predominate na microorganisms that kapag sila yung mas dumami ay mag-cause ng slime formation. We also have fat rancidity, and there are a small number of fat deteriorating microorganisms, but of course, oxidative rancidity is also possible, so prolonged exposure to high level of oxygen would cause fat rancidity in your meat and products. Of course, ito pinaka-common sa processed meats, or maybe if you have very high fat meat cuts. For mold growth, of course, you have your meats. So here are just some examples from Heinz and Kotzinger of what putrefaction looks like. This is, sabi natin, na greenish discoloration. In this case, kahit na processed meat na siya, yung mold growth. So, as we know, molds mas kaya nilang mag-grow under harsher conditions compared to bacteria. And of course, yung slime formation, baka hindi masyadong halata. Sure na ito. but I think you probably encountered slimy meat, o lalo na yung mga um, let's say dinefrost mo, kinuha mo yung part, tapos binalik mo sa freezer, tapos nag-defrost ka ulit, and then by the time na mo na yung last portion, well, posible na slimy siya. Alright, now let's look at infection versus intoxication. I gave a brief definition earlier, but let's have some examples, no? Again, as I mentioned, pag sinabi nating infection, kakainin mo yung mikrobyo ng buhay at kung ano man yung metabolic substances na ginagawa nila habang nasa tiyan mo na sila, yun yung mag-cause ng poisoning. On the other hand, intoxication is the ingestion of the toxins that were produced by the microorganisms beforehand, regardless kung buhay pa or patay na yung microbes na ingest in food. It's important to note the differences between microorganisms, especially in meat, in infection versus intoxication. So, for infection is usually Salmonella, E. coli, Listeria, and so on. While sikat na sikat for intoxication ay yung staph natin, Clostridia, and then of course toxin-producing molds. So here are examples. For example, salmonellosis happens when the population of Salmonella species are greater than 10 to the 5. Okay. Or for example, when E. coli O15787 is greater than 100 per gram, no? Kanilang plate count. And sikat ito noon, if you can remember yung mga na-food poison sa Jack in the Box US beef burger joint. And for intoxication, on the other hand, yung Staphylococcus aureus kasi marami siyang enterotoxin, and they're given letters. Staphylococcal enterotoxin letter E, yung isa sa mga pinaka-notorious. STB yung normal na tawag sa kanya. And this happens when you have an S. aureus plate count of greater than 10 to the 5 log per gram. Doon nagsisimulang mag-produce ng toxin yung staph. So it's the presence of staph doesn't necessarily mean na ma-intoxicate ka. Pero pag dumami siya at left unchecked, even if you were able to kill the staph cells, ay maiwan yung toxin. Then of course, we knew that botulism, meron itong mga outbreaks early 1920s before they finally understood the concept of thermal death time. And that's why we are very strict with canning, or especially our meat products. Kasi kapag hindi enough to kill the even the spores of these microorganisms, then they could proliferate inside the can. And hopefully nag-cause siya ng swelling or something para hindi makain yung laman kasi alam mo may mali na. Right? And usually parehas lang naman sila ng possible causes. Gastrointestinal disturbances, natin suka tae o lagnat. Worst case, may diarrhea ka, may dehydration, ikamatay mo yung anumang complications nun. And this is a real issue, especially in countries wherein food safety is almost non-existent. No? And even sa atin, sabi ko nga, ako hilig kong kumain ng mga isaw, pero I never eat isaw unless ako ay nasa bahay or malapit sa bahay, kasi alam ko na malaki yung chance na magkaroon nitong gastrointestinal disturbances after. Okay? So, yun. In terms of the time frame, most of the time frame varies from a few hours to more than a day, no? Pero usually ang intoxication syempre mas mabilis dahil andyan na yung toxin versus yung infection na pwedeng mag-party muna sa tiyan mo yung mga mikrobyo bago sila mag-produce ng metabolic substances. Okay? So, all in all, ang takeaway lang natin dito is kung na-infect or na-intoxicate, unang-una mahirap malaman kung alin kasi medyo pareho ng simptomas. Ikalawa, you have to trace back to probably up to 2 days before kung ano lahat ng kinain mo. Because it's not always the last thing you eat that caused your infection or intoxication. And lastly, medyo mahirap ma-determine yung root cause nito kasi kadalasan, bawa kung 24 hours ago yun, wala nang trace ng food sample para ipalab test. Unless halimbawa na i-ref mo yung food na hindi naubos, pwede mo pang check. And that's why ang hirap minsan ng mga outbreak to find the causative thing unless there's either enough of the sample or enough evidence to say sa interviewers na ano ba yung common na kinain nitong lahat ng nagkasakit sa isang party. Yung spaghetti pala, which is the number one cause of food poisoning in the Philippines. Diba? So, kasi yung mga hindi kumain ng spaghetti, hindi naman